I'm going to conclude my lectures on plant transformation by talking about plastid transformation. Plastid transformation offers many advantages for the molecular biologist over nuclear transformation, and those advantages stem from the prokaryotic-like nature of chloroplasts themselves. It's believed that plastids were introduced into a proto-eukaryotic cell in the dim and distant past, certainly at least 1 billion years ago, to produce a photosynthetic eukaryotic cell which has subsequently evolved into the flowering plants that we see nowadays. And there is excellent evidence for this. The first line of evidence is that this process of engulfment of cyanobacteria and their retention as symbionts within cells is still seen today in some marine animals and in some protists. And there is a lot of molecular evidence to support this theory. So the first is that plastid genomes are naked, covalently closed, circular DNA molecules devoid of histones, just like bacterial genomes. The replication of this plastid DNA is independent of the nuclear genome replication. And promoters of most chloroplast genes contain DNA sequences which are very similar to the E. coli 10' and 35' promoter motifs. Therefore, we can apply a lot of our understanding of prokaryotic bacterial DNA replication and gene structure to what's actually happening in the chloroplast. The chloroplast open reading frames are polycistronic. In the nucleus you have a promoter, you have a ribosome binding site, you have a gene, you have a poly-A tail. And that promoter drives the expression of that one gene. In bacteria, and in chloroplasts as well, the open reading frames are polycistronic. That is, you can have a number of genes expressed by a single promoter. So this leaves us with great potential for the expression. For example, if you want to express a whole enzyme pathway in the chloroplast, you just stack the genes up, put it behind a single strong promoter. And you can't do this with nuclear transformation. The plastid genomes contain few moderately or highly repetitive sequences. Plant genomes are huge. The smallest plant genome, Arabidopsis, is 50 million base pairs. Typical plant genomes are more like 1 billion base pairs. So if you're trying to put a gene into a genome, there's a lot of potential in plant nuclear genomes for recombination. There are a lot of homologous sequences where the gene can go in. So basically, when we do nuclear transformation, we don't know where the gene can go in. And this can mean that the gene can go in behind a strong promoter and be highly expressed, or it can go in behind silencer sequences and we get poor expression. So in any transformations that we carry out in the intact plant with nuclear transformation, we can expect a wide range in the expression levels. And basically we don't know where the gene is going into the genome. However, with plastid transformation, the genome is small, we know what it is to the nearest base pair, so we can put genes in by homologous recombination. So we know exactly where the gene is going into the plastid genome. And there are other similarities as well. For example, the chloroplast genomes of Euglena, Chlamydomonas and most angiosperms contain RNA, ribosomal RNA genes which are similar to their prokaryotic homologs. In terms of transcription, the RNA polymerases are very bacterial-like. I've already mentioned about operons and co-transcription. And the limiting regulatory step of gene expression is at post-transcriptional and translational level. So in the nucleus, a lot of the regulation is on transcription. So if you don't get messenger RNA being produced, then you end up with relatively low levels of messenger RNA and therefore protein. But in chloroplasts, you can have messenger RNA which sits quite happily in the chloroplast, and the levels of its regulation can be by post-transcriptional and post-translational. The transcription terminators are very similar to bacterial sequences, and chloroplast messenger RNAs tend not to be polyadenylated. Remember, in the nuclear genes we had to include a poly A tail. Transcription happens in the nucleus, but translation happens in the cytoplasm. So in order for the messenger RNA to get out of the nucleus into the cytoplasm, it had to be polyadenylated. It had to have this poly A tail on it. And it's not needed in the chloroplast. Plastid ribosomes are very bacterial in structure, and the ribosomal RNA gene sequences are very similar to modern cyanobacteria than to nuclear counterparts. So there's much more homology between chloroplasts and cyanobacteria than there is between cyanobacteria or plastids and plant nuclear genes. If you look at promoters, if you go to bacteria, you'll find the promoter elements around about 10 base pairs and 35 base pairs upstream of the start codon, and that's exactly what we find in chloroplasts. In purple in the middle here we've got a ribosome binding site, which is very similar to the Shine-Delgado sequences that you get in bacteria. And this is just demonstrating that chloroplast genes are actually organised into cistrons, where a single promoter can drive the expression of a number of different genes. Okay, so chloroplast translation, once the RNA has been transcribed, is very similar to prokaryotic translation. It makes use of 70S ribosomes. It uses 4-methionine as an initiator transfer RNA for the translation initiation codon. Messenger RNAs are not capped. Messenger RNAs are not polyadenylated. Like I say, there's no need for the gene to get out of the nucleus, therefore no need for polyadenylation. We have these Shine-Delgado-like sequences in the 5' untranslated region upstream of the DNA, of the RNA, and the RNA can actually exist as stable translational units.